Hi, my name is Natalie Chang. I would like to propose a project that looks for blood-based biomarkers for Alzheimer's disease. To give a bit of background about myself, I'm a junior at New Rochelle High School and my broad research interest is Alzheimer's disease. Over the course of sophomore year, I read journal articles in order to learn more about it. And over the past several months, I've really focused on blood-based biomarkers that can be used to aid in Alzheimer's disease diagnosis. Now let me tell you a bit about why I would like to conduct this research. Alzheimer's disease, which is a neurodegenerative condition, begins affecting the brain approximately 15 to 20 years prior to the onset of symptoms. This asymptomatic phase is called the preclinical stage, and due to the length of the preclinical stage, it is difficult to diagnose Alzheimer's disease in its early stages. Additionally, diagnosis relies on clinical observation and additional tests, and there's no definitive way to diagnose Alzheimer's except for in postmortem examinations. Some of the diagnostic tests that can be used are cerebrospinal fluid testing and brain imaging. For cerebrospinal fluid, amyloid beta levels and total tau, as well as phospho-tau levels, can be taken into account when diagnosing Alzheimer's disease. As for brain imaging, PET scans can be used to look for amyloid plaques and tau neurofibrillary tangles, while MRI can be used to look for brain atrophy as indicators of Alzheimer's disease. To bring all of what I just said together, the current progression of Alzheimer's disease involves a preclinical stage of 15 to 20 years in which there are no symptoms. By the time symptoms do emerge, significant neurodegeneration has already occurred. After an individual notices symptoms, they can be evaluated by a doctor who may use cognitive tests, cerebrospinal fluid testing, and brain imaging to aid in diagnosis. Once someone is diagnosed with Alzheimer's disease, they can begin receiving treatment, but they may not be a good candidate for clinical trials in which potential interventions may be most effective early on in Alzheimer's disease progression. An alternative to the current Alzheimer's progression would be to screen people early on, therefore allowing us to diagnose and begin treating people before the onset of symptoms and before significant neurodegeneration has occurred. If we're to screen people before symptoms emerge, we would have to rely on biomarkers. Current biomarkers, however, are unsuitable for screening, since cerebrospinal fluid collection is invasive, brain imaging is expensive, and both types of testing are unsuitable for general practice. An alternative to these tests would be blood tests, which look for blood-based biomarkers. It's important to note that even when symptoms do appear, it can still be difficult to diagnose Alzheimer's disease. This is because mild cognitive impairment, one of the early stages of Alzheimer's, is not exclusive to Alzheimer's disease. People with mild cognitive impairment, which is characterized by forgetting to attend events, poor judgment, and misplacing things frequently, may remain stable for an extended period of time or progress to a non-Alzheimer's disease dementia. Therefore, blood-based biomarkers could be used to screen people before the onset of symptoms or to definitively diagnose people with Alzheimer's when symptoms do emerge. I mentioned that blood tests and blood-based biomarkers could be an alternative to other tests and biomarkers, but why exactly would blood-based biomarkers be beneficial? Well, blood, which circulates throughout the body and interacts with the brain via the blood-brain barrier, contains a variety of substances that can be used as biomarkers, such as proteins and RNAs found in blood cells and exosomes. Additionally, blood tests, which are minimally invasive, easy to administer, and suitable for general practice, may be more cost-effective than other tests, given how next-generation sequencing has become increasingly more affordable. With that said, there are some challenges involving blood-based biomarkers. For instance, the blood-brain barrier hinders the travel of substances between the central nervous system and blood, which may make it difficult to use the blood to measure happenings in the brain. Additionally, biomarkers for Alzheimer's disease may be expressed by non-cerebral tissues. Therefore, it may be difficult to determine which results are due to Alzheimer's disease and which can be attributed to other factors. Other potential issues are that heterophilic antibodies may interfere with immunochemical tests, and protein biomarkers may be broken down by proteases in the plasma. A way to work around this last issue would be to look for non-protein biomarkers, such as RNAs. In summation, the problem is that there is no way to definitively diagnose Alzheimer's disease early on in its progression, be it in the preclinical phase when there are no symptoms, or the mild cognitive impairment phase, which is not exclusive to Alzheimer's disease. My goals are to identify blood-based biomarkers that can be used to screen for Alzheimer's in the preclinical phase and to indicate that someone with mild cognitive impairment will develop Alzheimer's disease. In order to address these goals, I looked for datasets from the Gene Expression Omnibus that had expression data taken from the blood. More specifically, I looked for datasets with samples from people who were cognitively normal, had mild cognitive impairment, and had Alzheimer's disease. The first dataset that I looked at contains high throughput data on the expression profiles of extracellular RNAs from plasma samples obtained from the same participants every few years. One of the 35 people who provided blood samples, all were 70 or older. 9 were controls that were cognitively normal throughout the sample collection period. 11 converted from cognitively normal to mild cognitive impairment during the collection period, and 15 had Alzheimer's disease throughout the collection period. This dataset allows for comparison to be made between people who converted from cognitively normal to mild cognitive impairment and age-matched controls who remain cognitively normal. While this dataset contains a lot of valuable information, it does not allow for the determination of biomarkers that may indicate that someone with mild cognitive impairment is going to progress to Alzheimer's disease as opposed to a different dementia. To address this gap, I looked for another dataset. The second dataset that I looked at contains array data on the microRNA expression profiles from serum. This dataset has expression profiles from people who had stable mild cognitive impairment and people who converted from mild cognitive impairment to Alzheimer's disease, thus addressing what the first dataset lacked. Having found 2 datasets, I had a basic idea that I wanted to do a comparison within each dataset and then look for overlap between the results of these 2 comparisons. In order to make sure that there would be any possibility of overlap, I first checked if the 2 datasets had any overlap between all of the microRNAs measured. The 2 datasets had different names for the microRNAs, with one using ensemble IDs and the other using version 21 MIR-based accessions. In order to see if there was any overlap between which microRNAs were measured, I first needed to convert the datasets to the same naming system. So I started off by using Biomark to convert the ensemble IDs in dataset GSE 136243 to MIR-based IDs. Next, I used the R package MIR-based converter to check what MIR-based version Biomark converted the IDs to, and I found that it converted them to version 21 or version 22. Following this, I used MIR-based converter to find the corresponding mature microRNAs for each of the MIR-based IDs, and I then used MIR-based converter to convert the MIR-based IDs to accessions. With the 2 datasets now using the same naming system, I compared them and found enough overlap to proceed. My proposed methodology involves using 2 datasets, GSE 136243 and GSE 150693 from the Gene Expression Omnibus. I plan to first work with GSE 136243, and my first step with this dataset would be to filter out all of the data on expression of non-microRNAs. Next, I will select samples from the participants who converted from cognitively normal to mild cognitive impairment provided before they converted. I will then look for differentially expressed microRNAs between these samples and the samples provided by the controls who remained cognitively normal throughout the collection period. Next, I will use GSE 150693 to look for differentially expressed microRNAs between the participants who converted from mild cognitive impairment to Alzheimer's disease and the participants who had stable mild cognitive impairment and did not convert to Alzheimer's. Following this, I will normalize the data obtained from the 2 analyses, since the data from the 2 datasets was produced with different assays. After this, I will look for overlap between the differentially expressed microRNAs found in the 2 aforementioned comparisons. And if I find any, I will annotate them through literature review to see what genes they may regulate and how those genes may be related to Alzheimer's disease. If I do not find any overlap, I will annotate the microRNAs identified in each separate comparison. Tying this back to the bigger picture, This research could contribute to finding blood-based biomarkers that could potentially be used for screening people for Alzheimer's disease prior to the onset of symptoms, or for definitively diagnosing individuals with mild cognitive impairment who are going to progress to Alzheimer's disease. In addition to having a diagnostic role, identified microRNAs may also be potential therapeutic targets because research suggests that microRNAs regulate various genes involved with the pathogenic pathways of Alzheimer's disease. I am very excited to have the opportunity to conduct this research, and I hope that under the guidance of a mentor, I will be able to produce valid results. I have read several journal articles in my field of interest and taken the coding courses and transcriptomics courses provided on the OmicsLogic site, and I look forward to applying what I have learned in this project. With that said, I would love to be able to consult a mentor if I have questions regarding sections of code and interpretation of statistical tests. To reiterate, my name is Natalie Chang, and I would like to look for blood-based microRNA biomarkers for Alzheimer's disease diagnosis. Thank you so much for your time.